Okay, so now we're going to look at sequencing, and we're going to start by comparing it to PCR because in both cases we're synthesizing DNA in a test tube. So in PCR, we needed template, primers, Taq polymerase, and DNTPs. We needed a lot of DNTPs because every PCR product is going to include many DNTPs, and you can't reuse them. Once you've used a DNTP, once you've incorporated it in a PCR product, it's gone. Same for the primer. Once you've used a primer to start building a PCR product, you don't get it back again. Taq polymerase is reusable. That's the reason we use Taq, is that it's heat stable. And template is reusable with an asterisk because the amount of usable template for the reaction doubles every cycle. Each PCR product from one reaction can be used as a template for the next round. That's why the CR in PCR stands for chain reaction. So we need a little bit of template, some Taq polymerase, a lot of primers, and a whole lot of DNTPs. So Sanger sequencing or cycle sequencing or chain terminator sequencing, sorry, they all mean more or less the same thing, uses very similar components. We still need template, we still need primers, but in this case we're using T3 primer or T7 primer. We're not using both. We're still using Taq polymerase, we're still using DNTPs, we're also using DDNTPs this time. Those are the dideoxynucleoside triphosphates. If you'll remember, they're missing a 3' OH. So when you incorporate a DDNTP into your sequencing product, synthesis stops. There's not a 3' hydroxyl for the 5' phosphate of the next nucleotide to attach to. So you incorporate a DDNTP just fine, but you can't incorporate anything after that. So looking at how much of what we're going to need, so the template is still reusable. The primers and DNTPs still aren't. DDNTPs are also not reusable. Taq polymerase still is. Those things are still the same. So as far as what you need the most of, it's still going to be it's still going to be DNTPs, because every sequencing product should hopefully incorporate a fair number of them. So you'll need a lot of DNTPs. As far as what you'll need the least of, that's actually still going to be template. The amount of template present doesn't increase the way it does in PCR, but we're still running this for many cycles. So you really only need a little template, comparatively speaking. You need more than for PCR, but you don't need all that much. So of all these components, you need the least amount of template. Then looking at the remaining 3 things we need. Every sequencing product is going to begin with a primer and end with a DDNTP. So the amounts of those that we use are going to be comparable. So the amounts of those that we put in are probably also going to be comparable. Taq polymerase, again, we run this for many cycles, and we can reuse the polymerase from cycle to cycle, whereas we can't the primers or the DDNTPs. So we need more Taq polymerase than template. We don't need tons, and we need more than that of primers and DDNTPs, roughly comparable amounts. So we need the least template, we need some Taq polymerase, we need more primers and dideoxy terminators, and we need the most just regular DNTPs to do the bulk of building the sequencing products from. And here's just a reminder, DNTPs versus DDNTPs. Neither of them have a 2' OH. DNTPs do have a 3' OH, DDNTPs do not. So you can't add another nucleotide after a DDNTP. It terminates the chain, thus the chain terminator sequencing name. These are the DDNTPs that we're using. I find it sort of amazing that DNA polymerase can incorporate these as if they were regular nucleotides, but apparently there's not steric hindrance from the fluorescent tags. Sort of blows my mind, but what do I know? So at the end of the sequencing reaction, we have all these different products of all different lengths. Remember, as we're adding nucleotides to the end of the primer, there's going to be a small chance of adding a dideoxy terminator and stopping, and a much larger chance of adding just a regular nucleotide, like we see in all these others, and continuing to synthesize. Then at the next position, small chance of incorporating a terminator, larger chance of incorporating a regular nucleotide and continuing, etc. So we're going to make, hopefully, the full range of possible sizes, ranging up to about 850 to 900 nucleotides long. And so at the end, we'll just have all these single-stranded pieces of all different sizes, each ending with a chain terminator with a fluorescent tag on them. So we need to separate them by size. So we do electrophoresis, this time in a capillary gel. And it's a type of gel that can separate a piece that's 62 nucleotides long from a piece that's 63 nucleotides long, which is kind of mind-blowing to me. But that gives us, it does give us clean separation between the products. And as those products go past the laser on the sequencing machine, they will glow the corresponding color, which will be recorded, and an algorithm will assign letters to that. This is what your sequence file output would look like. It's another of those things where there are many names for the same thing. It's a sequence file or a trace file or a chromatogram or an electropherogram. Sorry about that. They kind of all mean the same thing. We're mostly going to go with chromatogram or sequence file. But it's just showing you what color light flashed and how bright it was. And G's don't glow black, they glow yellow, but yellow is hard to see on computer screens. So common question is whether your chromatogram includes the sequence of the primer. And so something to think about there is recall that you don't start generating sequence. You don't start having the ability for something to glow a color until you've added a dideoxy nucleotide, or until you've had the chance to add a dideoxy nucleotide. So where's the first position that you could potentially add a dideoxy nucleotide? And is that ever going to give you the sequence of the primer? If you're not sure, ask your TA in block 2. But yeah, so just think it through in those terms. How do we get sequence? Where and when do we start having the chance to add dideoxy terminators to our product? So again, just looking at a generic recombinant plasmid, which is what we sent off for sequencing. You've got your T7 primer binding site, your T3 primer binding site in one tube. You'll be using T7 primer and sequencing your inserts starting from here, sequencing a little bit of pBlue script, and then going into the yeast DNA. In the other reaction tube, you'll be doing from the T3 side and sequencing that way. Hopefully, depending on the size of your insert, they might meet in the middle. If your insert is 2 kb or more, you might not be able to sequence the whole thing, but you can sequence enough of it to be pretty confident of what we've got. So we've got the sequencing going on in 2 separate tubes, the T3 tube, the T7 tube. So something to think about is why do we do that? Why do we have a T3 sequencing reaction and a T7 sequencing reaction? What would happen if instead we just put them both into the same tube? And so why don't we do that? And then of these 2 sequencing reactions, the T7 starts here and goes that way, the T3 that starts here and goes that way, should be relatively clear that your T3 sequencing reaction is more or less always going to go through the BAMH1 site to the EcoR1 site and into the genomic DNA. Your T7 reaction can also end up sequencing through this BAMH1 site if your insert is small enough. Again, we can generally get between 800 and 900 nucleotides worth of good sequence. So if your insert is smaller than that, your T7 reaction might also end up including the BAMH1 site on the other end, because they're just going to start and go for about 800 to 900 nucleotides. They don't know whether they're sequencing insert or vector or what. And at this point, go ahead and open up the sequencing related practice problems that are up on Sakai and work through those. And then during block 2, you'll have a chance to go over those with your TA.